Hi, my name is Leo, and today we'll be discussing the differences between the RNAi and the CRISPR systems for reducing gene expression. So RNAi, also known as RNA interference, is a reduction or knockdown of gene expression at the post-transcriptional level, whereas the CRISPR-Cas system is a permanent alteration or knockout of gene expression. RNAi targets mRNA, and the CRISPR system targets DNA. RNAi uh, has a it produces a hypomorphic phenotype, which is essentially a mutation that causes partial loss of gene function. And because it's only a partial loss, this could potentially produce off-target effects for whichever type of experiment you're trying to do. But an upside is that the cellular machinery will be conserved, which means that the cell will not die from this approach. Whereas the CRISPR system, it's a complete uh, alteration of the gene, which means that there are no uncertain effects that could be lingering. But it also means that it could be potentially lethal to the cell if the gene you're targeting is essential for cell function. So for this question, I would choose to use the CRISPR system because the goal of our study is to understand the effect of reducing or eliminating the protein production from gene A. And we're not given any other information about gene A. Other than that, no mutations have been noted. So the CRISPR system, it will target the DNA and completely nullify expression of gene A, which would lead to no protein production by gene A. And this will fulfill the requirements of our question because we will be able to see the effects of what happens after we completely nullify protein production. And while this is potentially lethal, uh, it would actually help us to obtain more information based on whether or not the cell continues to live or not after this method has been applied. And the RNA interference method, it's only a reduction, which means that there could be some effects that skew our results. But obviously, the most optimal approach would be to utilize both methods individually and compare and contrast the results at the end.